Hi students, moving ahead in the lesson sexual reproduction in flowering plants. Today we shall discuss the double fertilization and triple fusion and also the post fertilization changes and structural and events. Once the pollen tube enters inside the ovule to the embryo sac, it enters into one of the synergids inside the embryo sac. There, the tip of the pollen tube dissolves and the male gametes are set free into the cytoplasm. One of the male gamete moves towards the egg cell and fuses with the nucleus of the egg cell, completing the syngamy or the event of fertilization, leading to the formation of zygote, the diploid cell, and the other male gamete move towards the polar nuclei located in the center of the embryo sac and fuses with them to produce the triploid primary endosperm nucleus. This this event is called triple fusion, as the primary the secondary nuclei is containing the two female nucleus and one male nucleus with the male gamete. So three cells are fusing together, hence it is the event known as triple fusion, and this triple fusion results into the formation of primary endosperm nucleus, and the further it develops to form the endosperm tissue that is essential for the nourishment of the developing zygote. Here in the diagram 2.13, you can see here the development of the embryo after fertilization. This is the example shown in the dicot plant and the development of dicot embryo. In post fertilization changes, there is development of endosperm, and we have discussed how endosperm is formed. The endosperm is triploid as far as the ploidy of the endosperm tissue is concerned. The cells of this tissue is filled with reserve food material and is used for the nourishment of the developing embryo. The most common type of endosperm development undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. So, endosperm are obviously of a free nuclear type, either or of a cellular type. The number of free nuclei formed before the cellularization varies greatly. The coconut water formed from the tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel of the coconut is the example of cellular endosperm. The embryo that is formed at the micropylar end inside the embryo sac, where the zygote is situated. Most zygote divide only after certain amount of endosperm is formed, and this is an adaptation to provide assured nutrition to the developing embryo. Embryogeny, it is the process of development of embryo, and it is almost similar in both monocot and dicot. This figure 2.14 is depicting the structure of the typical embryo in a monocot, and the figure 2.13, diagram number B, is representing the embryo in the dicot plants. The zygote gives rise to the pro-embryo and subsequently the globular heart-shaped and mature embryo. A typical dicot embryo consists of embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledon is called epicotyle, which terminates with the plumule or the stem tip. The cylindrical portion below the level of cotyledon is known as the hypocotyle and terminates at its lower end in the radicle or root tip. Embryos of monocot plants possess only one cotyledon. In grass family, the cotyledon is called scutellum. That is situated towards one side of the embryonal axis. You can see here. This is scutellum in the diagram B, and scutellum is representing the cotyledon. In the center you can see here the shoot apex enclosed by coleoptile, and this shoot apex is nothing but the plumule, and below it you find here in the diagram B is radical, root cap and coleorhiza. This radical is going to form the root primordia, and below it is the root cap that is essential for the protection of the tender tissues of radicle inside the soil. So this is the structure of monocot embryo. That's all in this video. Thank you.